Chordoma is a very rare cancer with only about 300 patients diagnosed each year in the United States. Unlike most malignant tumors, chordoma is a slow-growing, locally aggressive, and invasive tumor. The current standard of treatment for chordoma is surgery. However, its location along critical anatomical structures makes clinical management difficult, and recurrence is a common event in patients, with a median survival of approximately 6 years. Therapeutic options for chordoma are limited due to the tumor's lack of sensitivity to both chemo and radiotherapy. Therefore, novel therapeutic strategies are needed to improve the overall survival of chordoma patients. Currently, chordomas can only be defined by histologic and immunohistochemical criteria. Very little is known about the molecular mechanisms that drive chordoma development, and there are no predictive or prognostic biomarkers for chordoma. In this review, we summarize the most recent findings in the field of chordoma genomics and epigenomics, from comparative genomic hybridization and fluorescence in situ hybridization, which examine chromosomal alterations, large-scale DNA sequencing to find gene mutations, microarrays to quantify mRNA and microRNA expression, and DNA-methylation profiling. These have led to significant discoveries towards the biological pathogenesis of chordoma and may potentiate a role for molecular therapy in the treatment of chordomas. High-resolution screening of genetic instabilities by CGH and FISH has provided a more comprehensive landscape of the chordoma genome and chordoma genesis. These studies have uncovered novel candidate oncogenes that are frequently gained in chordoma and novel tumor suppressor genes that are frequently lost. Large-scale DNA sequencing allows us to identify recurring single nucleotide polymorphisms, also known as SNPs, that may be involved in gene dysregulation and the development of chordoma. Microarray analysis of mRNA and microRNA expression highlights the significant differences in expression levels between chordoma tissues and cell lines and normal controls. DNA methylation profiling reveals the altered DNA methylation patterns that result in genetic instability or dysregulated gene expression, thus contributing to tumor genesis and malignant transformation in chordoma. These significant advances in identifying the molecular signatures of chordoma enable us to identify underlying tumor genesis mechanisms, such as cell growth, proliferation, apoptosis, and drug resistance mechanisms. It is clear that a link exists between chordoma development and these genetic and epigenetic alterations, including chromosomal rearrangements, DNA mutation and methylation, and mRNA and microRNA expression. In recent years, interest in chordoma has grown dramatically, and there have been important discoveries in chordoma biology, but further work is needed to understand the causes, molecular basis, and roles of these genomic and epigenetic instabilities in tumor cells. These integrative approaches may also provide an opportunity to identify diagnostic and prognostic biomarkers that can be applied towards the personalization of cancer therapy and significantly improve chordoma clinical outcomes.